What is the meaning of the cause of the disease? The underlying biochemistry, the underlying biochemical reaction, the defected biochemical reaction. So if we know the detail of this cause, we can easily diagnose, and then we can easily suggest the rational and the effective treatment of this disease. We can make screening tests available for early diagnosis, which will be helpful in monitoring the disease and progress of the disease, and ultimately lead to the therapy and response to the therapy. So biochemical investigation first of all started from the cause of the disease. We need to know the defected biochemical reaction. Then based on this defective biochemical reaction, we will suggest the effective treatment, and then by adding some screening test, and then ultimately lead to the treatment. So 4 things are there. First of all, the diagnosis, which leads to the proper diagnosis by screening tests, and then the severity of the disease, which leads to the treatment options. So this is the significance of biochemistry in medicine. Then coming towards the definition of the cell. As we know that cell is the physiological as well as morphological unit of life. It has a proper shape, it has a proper function. So every organ have different cells, but their functions vary according to the need of the different organs. So it could be defined, overall definition of the cell could be any of the protoplasmic mass making up the organized tissue. Means some cells are combined together to make up a tissue consisting of a nucleus and cytoplasm surrounding the nucleus. So this is the proper definition of the cell, which leads it's a protoplasmic mass which leads to the making up of the organized tissues, and these tissues make up the overall organ, and every tissue has a nucleus and a cytoplasm. So this is the diagram of the cell. Here you can see a different morphological unit, the overall shape of the cell. And as we have divided the cell into 2 main organs, it means the cell has a distinct separate organelle which contains most of the functions of the cell, which is the nucleus, and then the other part consists of cytoplasm. Some of the cytoplasm is cytosol and the other is cytoplasm. We will discuss in detail, but here you must know that the cell composed of 2 basic units, 2 basic divisions, which are the cell and the cytoplasm. Some of the detail of the different types of cells, as I have already told you that prokaryotic cells are of bacterial cells, and we only study the prokaryotic cell because of the fast replication of these prokaryotic cells, and we can do research easily on the prokaryotic cells, and due to the structural similarities between the prokaryotic and eukaryotic cell, we can easily make some of our diagnostic proteins, some of our treatment options from the prokaryotic cells. So some of the differences between the prokaryotic and eukaryotic cells, we can compare it by prokaryotic cells lacks a true nucleus, while eukaryotic cells membrane bound nucleus. Second difference is there is a single chromosome present in case of prokaryotic cell, while eukaryotic cells composed of 1 chromosome. The overall chromosome, which is further by zooming it in, We can see the DNA on this chromosome, while in case of prokaryotes we have two types of chromosomal material. One is chromosome and one is the extra chromosomal material, that is known as the plasmid. Example of the prokaryotic cell is bacterial cell, while that of eukaryotic cell is animal cells, plant cells, fungal cells, etc. So you must know 2 to 3 differences between the different types of the cell. Now coming towards the details of the chemical composition and functions of the cell. Cell is grossly divisible into nucleus and the cytoplasm. Here you can see some of the cells of one tissue where you can see the nucleus and the cytoplasmic region. So overall division of the cell is nucleus and the cytoplasm. What is cytoplasm? Cytoplasm is the part of the cell other than the nucleus. It is basically composed of aqueous solution, which is called cytosol. So we can say that the part of the cytoplasm which is aqueous in nature, which is liquid in nature, this is known as cytosol. A variety of suspended particles and organelles like mitochondria, endoplasmic reticulum, free ribosomes, Golgi bodies are also present in the cytoplasm. Starting from the nucleus, then we will discuss the different components or different organelles of the cytoplasm. Nucleus is the central core of the cell. So if we are going to study the cell, first of all we have to study the nucleus. Nucleus is the central core of the cell. It has one or more nuclei. It has one or more nuclei. It is also composed of a nucleoplasm, nuclear membrane, and nucleoporins. So the nucleus is subdivided into different components like nucleoplasm, nuclear membrane, and nuclear porins. What are these? We will see it individually. This is the structure of the nucleus. The nucleus is made up of chromatin, and here you can see the nuclear envelope, the outer covering of the nucleus, nuclear pores like nucleoporins, which are protein in nature and responsible for the transportation of different biomolecules, and the chromatin material is present in case of nucleus, which could be euchromatin and heterochromatin. Let's see its detail. Nucleus, it contains chromosomes and chromatin. Chromatin composed of DNA and proteins. You know what? DNA is a thread-like structure. DNA cannot be folded on itself. So DNA needs some special types of proteins for the folding of the DNA onto it. So these special types of proteins are known as histone proteins. When the DNA bound on these histone proteins, it makes two types of structure. When the DNA is not bound to the histone protein, it is known as euchromatin. Means DNA is free from the histone proteins and is ready for the replication process. While in case of heterochromatin, DNA is condensedly packed around the histone proteins. So the other names of euchromatin and heterochromatins could be true chromatin and false chromatin respectively. So we can say that in the chromatin structure is divided into euchromatin and heterochromatin. When the cell needs to replicate itself, it needs to be converted into euchromatin type so that the different processes could take place on it. When the processes are over, then the euchromatin is turned back into heterochromatin. It is again packed on the histone proteins. So what are the two different types of chromatin? These are euchromatin and heterochromatin. Euchromatin, the name for the euchromatin is true chromatin, means it is ready for the division, and false chromatin is not ready for the division. Then coming towards the details of the euchromatin and heterochromatin. Euchromatin is transcriptionally active, less condensely packed or uncondensed. Heterochromatin is transcriptionally inactive, densely packed during interphase. It's condensed. And from the euchromatin and heterochromatin is further divided into two types, constitutive and facultative. The part which is constitutive in heterochromatin, it is always condensed and thus essentially inactive. It is found in the regions near chromosomal centromeres or telomeres. The other part of the nucleus, heterochromatin, which is facultative, it is condensed at times, but at other times it is actively transcribed and thus uncondensed and appears as a euchromatin. So some of the DNA also contain non-histone proteins, which help in maintaining the chromosomal structure. It regulates the DNA transcription. For example, DNA polymerases are non-histone proteins. RNA polymerases, in fact, these are enzymes, but all enzymes are proteins in nature. The proteins other than the histone proteins are known as histone proteins, which could be DNA polymerases and RNA polymerases. So you can define them on the basis of their functions, like polymerases always helps in the DNA replication, and RNA polymerases are helpful in the transcription processes. So major functions of the nucleus is genetic information of the cell. Genetic information of the cells means the DNA is first replicated and then transcribed and then translated, which is all started from the nucleus and then completed in the cytoplasm by completing the phenomena of central dogma of molecular biology. What is this? The complete set or completion of the different processes like transcription as well as translation. Now coming into the details of the nuclear components, it also contains micronucleus, concerned with the synthesis and assembly of ribosomes, site of ribosome biogenesis. It is not surrounded by a distinct membrane. Here you can see the thread-like structure of the nucleus. When it got bounded around the histone proteins, it made up the chromatin. So this is all about today's lecture. From tomorrow we will start discussing the different organelles which are present in the cytoplasm. Thank you.